In this lecture, we're going to revisit some ideas that we learned in Lecture 2 to review, because I understand it was a lot of information that was given to you. And we're going to review some concepts that we haven't discussed in a while. So we're going to talk about how respiration relates to things like surface area and pH and evolution. We are going to tackle a few new concepts today related to anaerobic and aerobic respiration. So first, let's talk about respiration and its relation to surface area. Now, every student should have memorized at least one idea about how an increase in surface area can increase the efficiency of a cell. And a good example to discuss would be the folds in the inner membrane of the mitochondria. Now, these folds have a name. They're called the cristae. And this is what I mean by the cristae. So this fold here of this inner membrane increases the surface area where oxidative phosphorylation can occur. Now, one thing I want to point out is that on a previous page, we drew in the electron transport chain embedded in the inner membrane of this mitochondria, but I only drew one, but there's many of them. So let's indicate that by putting some more electron transport chains in there and more ATP synthase molecules. And so if we have more folds in the membrane, then we have a greater surface area where these important proteins can be embedded. So greater surface area is going to lead to a greater amount of ATP production. In the biochemistry unit, we talked about acids and bases, and we said that the acidity of a solution is measured by the hydrogen ion concentration. And if you remember, a greater concentration of hydrogen ions, which are protons, will result in an environment that is more acidic. So let's talk about this idea in relation to the mitochondria, and I'm going to go back a little bit here. So remember in oxidative phosphorylation, as electrons, so if we want to put an E here, as electrons are being moved down the electron transport chain, these proteins are moving protons or hydrogen ions into that inner membrane space. We're getting a high concentration of protons. And these protons are going to move down their electrochemical gradient. So they're going to flow through that protein, ATP synthase. And in doing so, that gives ATP synthase energy to produce ATP molecules. Now, because of this high proton concentration in that inner membrane space, the inner membrane space of the mitochondria is more acidic. It's more acidic compared to the matrix of the mitochondria. The matrix of the mitochondria has a lower hydrogen ion or proton concentration. So since the inner membrane space is more acidic, then we know that it's going to have a lower pH compared to the matrix. Now let's relate this idea of pH to hemoglobin and oxygen transport. This diagram is showing you what is taking place at the lungs and taking place in other tissues of the body. So blood is dropping off carbon dioxide into the lungs and you're exhaling it. So technically there's less CO2 in the blood at the lungs. Now, if you remember, if there's less CO2, then there's going to be less carbonic acid in the blood, and so our blood is going to be more basic. And because the blood is more basic, hemoglobin has an altered shape, which increases the affinity for oxygen. That means that the hemoglobin is going to bind more oxygen whenever the blood is more basic compared to acidic. So here we have oxygen moving into the bloodstream, but let's show those oxygen molecules being attached to hemoglobin inside the red blood cells. So I'm going to draw these circles to represent the red blood cells that contain the hemoglobin that are carrying the oxygen. Whenever the red blood cells arrive at the tissue, that tissue is performing respiration. It's making ATP, and remember, it is also making CO2. So as that CO2 moves into the fluid surrounding the tissues and finally into the bloodstream, then we're going to get a drop in pH. Our blood is going to become more acidic in these areas. That, again, is going to alter the shape of hemoglobin, and hemoglobin is going to have less of an affinity for oxygen. That means that it's able to release the oxygen to these tissues that truly need the oxygen. So let's indicate that low CO2 levels result in blood that is less acidic, and hemoglobin has a greater affinity for oxygen. So it's going to bind to oxygen in the lungs. Tissues are going to have more of an acidic condition. Since tissues are making ATP and producing CO2, then the fluid around those tissues is going to be more acidic. And hemoglobin has a lower affinity for oxygen in that condition. And again, this is just making sure that hemoglobin is delivering oxygen to those tissues that have high ATP demands, which means they have high oxygen demands and are going to produce more CO2. Now, let's discuss ATP production in bacteria, because all cells, plants and animal cells, bacteria cells, fungi, all utilize ATP to provide energy for their reactions. But if you remember, bacteria are prokaryotic cells, which means they have no membrane-bound organelles. So that means they do not have mitochondria. How are they able to meet these ATP demands of the cell? So let me move down here so you can see this diagram that I have of a bacteria cell. I imagine the first idea that jumps into your head when you look at this diagram is that this bacteria cell looks a lot like the mitochondria. Just like the mitochondria have created this space, which is called the intermembrane space, bacteria have a space as well, and it's called the periplasmic space. As you can imagine, because there's a space there, that gives this bacteria cell an area to concentrate protons or hydrogen ions. Now remember, bacteria cells have a wall, and some even have two membranes. They have an inner plasma membrane, and they also have this outer membrane. And it's because of these structures that we get this periplasmic space created in these bacteria cells. Now, just like your cells utilize glycolysis to break down glucose, bacteria cells utilize glycolysis as well. And they can also oxidize other molecules like lactose and amino acids. And we'll talk more about those in a second, because we can also oxidize those as well and get energy from those types of molecules. But let's just indicate that in the cytoplasm, since this is just one cell, this is a bacteria cell, glycolysis is occurring. So let's take glucose, and in the process of glycolysis, we get two molecules of pyruvate. And remember, in the oxidation of glucose, we're removing hydrogen ions from some of these intermediates, and we're placing them on NAD. And that reduces NAD to NADH. Now, these pyruvates will enter the Krebs cycle. And I'll clarify that pyruvate is converted to acetyl coenzyme A, and it's acetyl coenzyme A that enters the Krebs cycle. So let's indicate the Krebs cycle is occurring here in the cytoplasm of these bacteria cells. And remember, the Krebs cycle is also oxidizing these intermediates, placing electrons on NAD to form NADH. And now we have these proton and electron carriers that are generated in these bacteria cells. Now, just like in our cells, these proton carriers or electron carriers are going to drop off these protons and electrons at the electron transport chain. So you can see here in the inner membrane of these bacteria cells, we have electron transport chains. We have proteins that are embedded. So let's indicate these NADH molecules are going to release their hydrogens or their protons, and they're going to release their electrons. So let's show an E, and these electrons are passed down the electron transport chain. As they're passed, hydrogen ions are being pumped into that periplasmic space. And again, we're getting a high concentration. We have an acidic environment there. And these protons are going to float down their electrochemical gradient back into the cytoplasm. And in the process, ATP synthase, which you can see right here, is going to phosphorylate ADP. Now, the majority of bacteria cells require oxygen, just like you require oxygen, and the oxygen is required right here. It is the final electron acceptor. So oxygen is going to accept that electron off of the last protein in the electron transport chain. It's also going to combine with two hydrogens, and it's going to form water. Okay, so let's indicate up here that bacteria cells are utilizing glycolysis in the Krebs cycle, and they're also using oxidative phosphorylation. They are reducing NAD plus to NADH, and then we're using those protons in the establishment of this gradient to produce ATP. Now, on this page, I want to revisit that idea that I mentioned on the previous page, that bacteria can utilize glucose and lactose and amino acids to oxidize so that we can get energy, or we can make ATP. And I want to point out that your cells can utilize those molecules as well. So these are not diagrams that you need to have memorized, but you should be able to look at this and indicate this big idea that they want you to know, is that there are many different molecules that we can obtain energy from. So energy can be obtained from amino acids, from the proteins that we eat, and fats. And these different molecules can enter these aerobic processes at different points. Now, you might not be too familiar with names of amino acids yet, and you'll become more familiar with them when we get into protein synthesis. But these right here are amino acids. So if you want to indicate those are amino acids, and so are those, and so are those. And it's simply showing you that these amino acids can be converted into some of these intermediates, like acetyl coenzyme A or pyruvate. And again, these molecules are important molecules in respiration. Over here, it's showing you that if we break down fats, remember triglycerides contain a glycerol section, and then they have fatty acids. So these fats can be metabolized, broken down, and the glycerol and these fatty acids can be converted to some of these intermediates in aerobic respiration. Again, you don't have to have all this information memorized, but I would try to memorize one key point that you can utilize as an example. I think the easiest thing to remember is that some amino acids can be converted to acetyl coenzyme A. I'm talking about right here. But also, fatty acids can be converted into acetyl coenzyme A as well. So I think that's an easy molecule to remember. So remember acetyl coenzyme A, and we can convert many molecules, amino acids and fatty acids, to acetyl coenzyme A. Since some amino acids and some fats can be converted to acetyl coenzyme A, I think that is a good fact to have memorized. So let's indicate some amino acids and fatty acids can be converted to acetyl coenzyme A that is utilized by the Krebs cycle. Now let's talk about other sugars, because we can get energy from glucose and fructose, and lactose can be broken down into galactose and glucose. So other sugars can provide energy as well. So let's indicate glycogen can be broken down into glucose subunits. So glycogen can be broken down, and here's a glucose subunit that we get from glycogen. And so that can enter glycolysis, galactose, and you can see here that when we break down lactose, which is milk sugar, then we end up with a molecule of galactose, and it can enter this process of glycolysis. Fructose can enter here, and it's not necessary that, again, you have it memorized that fructose can be converted to fructose 6-phosphate. So you do not have to have those details memorized, but the big idea to understand is that there's multiple energy sources, not just glucose, and that these different molecules can enter these pathways in different areas of respiration. Now, I have yet to mention anaerobic respiration. We've mostly been talking about aerobic respiration. Now, if you're doing aerobics, your instructor is going to want you to maintain a level where you are able to get in enough oxygen and deliver that oxygen to your tissues. And that's why that type of exercise is considered aerobic. So aerobic means requires oxygen. Now, if I ask you to summarize what takes place in the steps of aerobic respiration, then you're going to discuss all 4 processes of respiration like we did in the previous lecture. You're going to start with glycolysis and then talk about the prep reaction, the Krebs cycle, and then oxidative phosphorylation. So as a whole, these 4 steps are collectively called aerobic respiration. But one thing that I want to point out is glycolysis itself does not require oxygen. You can go back and look over the diagram that shows glycolysis and you will never see an oxygen molecule being utilized. So technically, glycolysis is considered an anaerobic process. So I wanted to point that out. But remember, it is a part of aerobic respiration because glycolysis has to happen first before the prep reaction, the Krebs cycle, and oxidative phosphorylation can take place. In some cases, when we're out of shape or we are exercising harder than usual, our cells can run out of oxygen. Our body is not supplying oxygen as fast as the cells are utilizing it. So our body can switch over and do what's called anaerobic respiration. Now, if I ask you to summarize the steps involving anaerobic respiration, you're going to discuss glycolysis again. And the glycolysis and the anaerobic process looks just like glycolysis and the aerobic process. But the next step after glycolysis is not the prep reaction like we have with aerobic respiration. It's the fermentation step. And glycolysis and fermentation are both considered anaerobic processes. They do not require oxygen for these processes to occur. So this diagram is showing you that first glycolysis will occur in those cells. And then the second step, if we want to add it here, is fermentation. And again, this is showing you that when there's no oxygen available, your cells are going to do fermentation instead of the prep reaction, the Krebs cycle, and oxidative phosphorylation. And in pre-AP, we learned that there's 2 types of fermentation. There's lactic acid fermentation, and then there's alcohol fermentation. And we talked about we do lactic acid fermentation, and that's why we are sore after a hard practice. And we also envision what life would be like if we didn't do lactic acid fermentation, if we instead did alcohol fermentation. So that would probably lead to some pretty crazy practices, especially at the beginning of the season when everyone is out of shape. Now, before we take a closer look at these fermentation steps, I need to indicate why we have to have a backup plan when there's no oxygen. If oxygen is not present, then the Krebs cycle and oxidative phosphorylation they stop. And you need to understand why these 2 processes stop. So let's indicate that no oxygen is here to be the final electron acceptor. So this electron cannot be removed from that protein and electron transport chain, and this one cannot move, and this one cannot move, and so on. And since there's an electron here, FADH2 and NADH are not able to unload their electrons. So we're getting this high production of NADH and FADH2. So let's indicate that we now have a bunch of these carriers, and they're being made, but they're not able to release those protons and those electrons. Now remember that to make NADH and FADH2, you have to have FAD molecules and you have to have NAD molecules. And if they're all tied up with protons and electrons, then there's no more FAD and NAD for the citric acid cycle to occur or the Krebs cycle to occur. So that's why the Krebs cycle stops, is because it runs out of FAD and NAD because all of it has been reduced already to NADH and FADH2, and yet those molecules aren't able to donate their electrons here to the electron transport chain. Now, of course, the electron transport chain is going to stop simply because we are not able to move these electrons down the electron transport chain because there's no oxygen here to act as that final electron acceptor. So if electrons aren't being moved, then we're not going to have this high concentration gradient. So that means that there is nothing to flow through ATP synthase, and we're not going to get any ATP. There's always a possibility that you would have to indicate why the Krebs cycle and why oxidative phosphorylation stops. Now I want to spend a second to talk about anaerobic and aerobic respiration and to compare these two processes. So this diagram is summarizing the main difference between aerobic and anaerobic respiration is that after glycolysis occurs, and again glycolysis occurs in both processes, if oxygen is present, then we're going to get the prep reaction occurring. So we're going to get the conversion of pyruvate to acetyl coenzyme A. The citric acid cycle will occur, and then oxidative phosphorylation is going to occur. So if we want to put some electron transport chain proteins in there, an ATP synthase, and show that these reduced NADH molecules are going to drop off their protons and their electrons so that we can concentrate these protons so that we can generate ATP. Now, if oxygen is not present, then the second step after glycolysis is going to be fermentation. And this diagram is showing you that there's two types of fermentation. We can get ethanol produced, and that's alcohol, or we can get lactate produced, or lactic acid. So a second type of fermentation is lactic acid fermentation. So let's indicate that in some of these blanks, and then we're going to go back up to the diagram again. So again, anaerobic respiration is going to occur whenever the cell is not getting enough oxygen. And there's two steps. First, glycolysis occurs. And as you can see from the diagram, the glycolysis in aerobic respiration and anaerobic respiration is exactly the same. But that second step is different. Instead of the prep reaction, if there's no oxygen, then the next step is the fermentation step. Again, there's two types of fermentation. Animals and some bacteria are capable of doing lactic acid fermentation when they're lacking oxygen. And yeast, which is a fungi, when they're lacking oxygen, they will switch over to alcohol fermentation. Now, on this diagram down here, I want to show you just the big ideas that are occurring in these two steps, glycolysis and fermentation. So again, in glycolysis, glucose or other molecules are being converted to pyruvate. And in that process, if you remember, we end up with 4 ATP being produced with a net of 2. We're also generating NADH molecules. So let's show NAD being reduced to NADH. Now remember, whenever there's oxygen, these NADH molecules are being sent to that last step, oxidative phosphorylation. But this process can only occur whenever there are oxygen atoms to be the final electron acceptor. So we cannot send these NADHs there, so let's indicate that. And as glycolysis occurs, we're going to get a buildup of NADHs. Now, if you remember, the Krebs cycle stopped working because eventually there was no more NAD plus molecules and no more FAD plus molecules to continue to oxidize those intermediates in the Krebs cycle. And that's what's going to happen to glycolysis here, is that we're going to run out of NAD+. So we're no longer going to have this supply of NAD+, so that glycolysis can occur to make 4 ATP, because these NADHs have been produced, but they're not being oxidized at that last step, oxidative phosphorylation. So this is where fermentation comes in. And in fermentation, pyruvate, which is the molecule that's produced at the end of glycolysis, is going to be converted to lactic acid or alcohol. So this step is showing you what is taking place in the fermentation step. Fermentation is going to help out this process of glycolysis by re-giving it or regenerating some NAD+. So in fermentation, these NADH molecules are going to be oxidized. They're going to donate their hydrogens and their electrons to pyruvate. And in that donation, we're getting the creation of lactic acid or alcohol. But this is really the important part, is these NAD plus molecules are being regenerated. So now we have some NAD plus molecules that can be utilized in glycolysis. And as long as glycolysis is running, then the cell is at least getting 4 ATP molecules. It's not getting a lot, but at least it's getting some. So I indicated down here that glycolysis is occurring, but if the cell is out of oxygen, then these NADHs that are produced are not going to be oxidized. So the purpose of this fermentation step is to oxidize NADHs, creating those NAD plus molecules that can be utilized in glycolysis. So at least the cell is getting 4 ATP per glucose molecule in the absence of oxygen. And I just want to re-show you that on this diagram, is that glycolysis is producing these NADH molecules. And again, normally they are utilized here in oxidative phosphorylation. They'll release, or they'll be oxidized there, releasing their protons and their electrons, so ATP can be generated. But if there's no oxygen present, oxidative phosphorylation shuts down. And so we're going to get a buildup of NADH. So the fermentation step is a way to regenerate this NAD that is needed so that glycolysis can occur, so that the cell can at least produce 4 ATP molecules per glucose. So the NADH is going to be oxidized into NAD+. In the process of reducing pyruvate, because remember, pyruvate is going to be accepting these hydrogens and these electrons, then we get the formation of these different molecules, ethanol or lactic acid. Now again, you don't have to have all of these steps memorized. You should not have to completely recreate every bit of these diagrams. These diagrams are showing you alcohol fermentation and they're showing you lactic acid fermentation, but you should be able to summarize why fermentation is occurring, and in general, what is the purpose of fermentation. So again, if a cell is doing alcohol fermentation, then it's most likely a yeast cell, and yeast cells are doing that because they have to be able to regenerate NAD+, so that glycolysis can occur to produce ATP molecules. So this diagram is showing you glycolysis, step 1, and then over here is step 2. This is where it's showing you the fermentation process. You can see that the pyruvate molecules are eventually going to be reduced, forming ethanol or alcohol. Now what might throw you off a little bit is, first, pyruvate has some carbon atoms removed, and now it's a molecule called acetaldehyde. And eventually the acetaldehyde is going to be reduced, and you can see it's being reduced because these NADHs are going to donate their protons and their electrons to acetaldehyde, and you can see the acetaldehyde has picked up a hydrogen atom right there, and you can see that it used to not have that. So don't get caught up with talking about acetaldehyde. You can just simply say that during the fermentation step, pyruvate from glycolysis is reduced to ethanol. So that means that it's going to accept those protons and those electrons. That NADH is oxidized whenever it's donating that to pyruvate, and this is so that the cell can regenerate that NAD+. And you also see that since those carbon atoms are being removed from pyruvate, that CO2 is produced in this process. Now, because yeast make alcohol when they're deficient in oxygen, then we can utilize them to make alcohol. And we can also use yeast to make bread. And that's not really due to alcohol fermentation. It's just simply whenever the yeast are performing aerobic respiration or even anaerobic respiration, then they're producing CO2 gas, and that is causing the dough to get these air pockets, and that's why the dough raises. Now the other diagram is just showing you lactic acid fermentation, and again, you're seeing glycolysis is the first step, and we're getting ATP generated, a net of 2. We're also generating these NADH molecules, but they're not being utilized in oxidative phosphorylation. So to regenerate NAD+, then fermentation is going to occur. So this is showing you the fermentation step over here. So here we have pyruvate, and you can see that it is directly reduced, and you can see that it's gaining this hydrogen right here, and it's accepting it from NADH there. So this is the process that allows animal cells and some bacteria cells to regenerate their NAD so that the cell can produce at least 4 ATP in glycolysis. And so we say that pyruvate is being reduced to lactic acid, NADH is being oxidized to regenerate that NAD. Now, notice that there's no release of carbon atoms. So this process does not release CO2. So I put a line there to try to help you remember that only alcohol fermentation releases CO2. And since we do lactic acid fermentation, that lactic acid can build up in our muscles, and it can make our muscles feel sore for about 3 days. And then I've been mentioning that bacteria cells can do lactic acid fermentation. This is the name of a bacteria cell, Lactobacillus acidophilus, and we utilize it to make yogurt because we will mix it with milk, we will deprive it of oxygen, and when it makes the lactic acid, it releases the lactic acid into the milk, and that acid causes milk to curdle. Just kind of in the same way that if you take vinegar and you mix it with milk, you're going to get milk to curdle as well, as those proteins in the milk are changing shape due to the change in acidity. So we can use the fact that they produce lactic acid in the absence of oxygen to thicken up milk, making yogurt. Let's relate respiration to evolution and this big idea that the process of evolution drives the diversity and the unity of life. If you remember, an idea underneath that is that commonalities between different organisms suggest a common ancestor. And one commonality that we have between all organisms is that they perform glycolysis. Scientists believe that glycolysis was performed by the earliest prokaryotes, and since that was a beneficial process, it was selected for over time and was passed down from generation to generation. Something that we know is that atmospheric oxygen did not exist early in the Earth's development. And so this fits with glycolysis because glycolysis is a process that does not utilize oxygen. It is an anaerobic process. So this process could have developed during the Earth's early history, whereas oxidative phosphorylation wouldn't have developed early in the Earth's history simply because the Earth did not contain atmospheric oxygen. We also know that glycolysis does not require mitochondria, and the Earth's earliest prokaryotic cells would not have possessed mitochondria, and it occurs in the cytoplasm. In the cell unit, we talked about the similarities between bacteria and mitochondria and chloroplast. And we have this idea that those organelles had ancestors that were once free-living prokaryotic cells, and that these organelles arose due to endosymbiosis. And just like any theory, the theory of endosymbiosis has to be backed by evidence. And we discussed in the last unit that both mitochondria, chloroplasts, and bacteria, they all contain circular DNA, they have similar ribosomes, and you can see them here in this diagram of the mitochondria. All three of them elongate and divide by binary fission. And you can also see that they're relatively the same size. So there's many similarities between bacteria cells and mitochondria and chloroplasts.